What if you could rewrite the book of life? I mean, really rewrite it. Not just read our genetic code, but actually go in and edit it. Correcting the tiny typos that cause devastating diseases, maybe even changing the entire course of human evolution. Well, this isn't science fiction anymore. It's the reality of a discovery that has changed absolutely everything. And the story starts with one incredible scientist. This is Jennifer Doudna. And in 2020, she and her collaborator, Emmanuelle Charpentier, received the Nobel Prize in Chemistry. We're talking the highest honor in science. And the award was for a breakthrough that many are calling one of the most important discoveries in the history of biology, period. The official citation from the Nobel Committee sounds almost simple, right? For the development of a method for genome editing. But the implications behind those words are just monumental. They figured out how to create a tool so precise it's like a pair of molecular scissors for our DNA. You might have heard of it. It's called CRISPR-Cas9. So you have to wonder, how does this happen? To really get a handle on this revolution, you've got to understand the revolutionary herself. And Jennifer Doudna's story is just as amazing as her science. It's a journey that takes us all the way from the islands of Hawaii to the most famous labs in the world. Every great scientist has an origin story. A moment where that initial spark of curiosity just ignites. For Doudna, that happened really early on, growing up surrounded by the incredible nature of Hilo, Hawaii. I mean, think about it. This place was her own natural laboratory, full of strange sleeping plants and hidden waterfalls. It made her wonder, how does life actually work? You know how sometimes one little thing can change the entire course of your life? Well, for Doudna, it was a book. When she was 12, her dad gave her a copy of The Double Helix, and she said it wasn't just some science story. To her, it read like a detective novel. It was a puzzle about life's biggest secrets, and right then and there, she decided she wanted to be one of the detectives trying to solve it. And that inspiration, it lit a fire under her. You can see it in her trajectory. She dove headfirst into biochemistry at Pomona College, then went on to get her PhD at Harvard Medical School, where she started to specialize in the super complex world of RNA. Every single step was another brick in the foundation she'd need for the massive discovery that was coming. But here's the thing. It wasn't a straight line to the Nobel Prize. Not at all. Like a lot of us, she hit a point of really serious self-doubt. A moment that, honestly, could have changed the course of modern biology forever. It's kind of wild to think about, isn't it? She was really struggling with chemistry and was this close to switching her major to French. But in one of those conversations that changes everything, her French teacher actually told her to stick with science. It just goes to show you, sometimes a little bit of encouragement at the right time can make all the difference in the world. Okay, so a breakthrough like CRISPR doesn't just happen in a flash of inspiration. It was really the peak of a mountain built from decades of painstaking fundamental research into one of DNA's less famous but super important cousins, RNA. See, Doudna's early work was all about these fascinating little things called ribozymes. Before her, the dogma in biology was that only proteins could be enzymes, the molecules that speed up all the reactions in our bodies. But the discovery of ribozymes proved that RNA could be a workhorse too, which opened up a whole new way of thinking about biochemistry. So she spent years trying to see the invisible. She used this incredibly powerful technique called X-ray crystallography. Think of it like creating a 3D blueprint of a molecule. And she was one of the very first people to figure out the exact shape of these RNA enzymes. This deep, deep understanding of how RNA folds and works, well, it turned out to be the exact expertise the world needed for what was coming next. And that brings us to the big moment, 2012. After all those years focused on the nitty-gritty of RNA, Doudna's work suddenly crashed into research from a totally different field, how bacteria fight viruses. And together, they created a technology that would change the world. So scientists had figured out that bacteria have this ancient defense system called CRISPR. You can think of it like a bacterial immune system. It basically keeps little snippets of DNA from invading viruses, kind of like a most wanted list. So if that virus attacks again, a protein called Cas9 uses a guide RNA to find the matching viral DNA and just chops it to pieces. Doudna and Charpentier's incredible insight was realizing they could hijack this system and reprogram it, turning it from a virus killer into a gene editor that could work on any DNA. And the impact was just immediate and staggering. Before CRISPR, trying to edit genes was, well, it was like trying to edit a single letter in a book with a sledgehammer. It was incredibly difficult, it was slow, and it was ridiculously expensive. This discovery turned it into a process as easy and precise as using find and replace in a word processor. It basically democratized gene editing for labs everywhere. The way it works is just so elegant. First, you create a guide RNA that matches the exact genetic sequence you want to change. That guide leads the Cas9 protein, the scissors, to that precise spot. Then, snip. Cas9 makes a clean cut. Finally, the cell's own natural repair crew comes in to fix the break. And here's the genius part. You can give that repair crew a new piece of DNA to use as a template, tricking it into writing your new code right into the genome. It's find and replace for the code of life. Honestly, the best way to think about it is like a biological GPS. The guide RNA is the address you type in. The Cas9 protein is the car that drives directly to that location and makes the change. It's this programmability that makes it so revolutionary. It's precise, it's efficient, and it has changed absolutely everything. But when you create a tool this powerful, it doesn't just stay in the lab, right? It immediately explodes into the real world. And that brings up some huge questions about its incredible promise, but also the profound ethical problems it creates. The promise is, well, it's breathtaking. We're talking about the potential for one-time cures for thousands of different genetic diseases. Things like sickle cell anemia, cystic fibrosis, Huntington's disease. Conditions that used to be a life sentence are now the targets of CRISPR therapies that are actually in clinical trials today. But with all that promise comes some serious peril. And Doudna herself became one of the first to sound the alarm, saying, "Whoa, hang on, we need to talk about this." She drew a really crucial line in the sand between two types of editing. Somatic editing fixes genes in a specific patient's body. Those changes die with them. She's all for that. But germline editing alters genes in sperm, eggs, or embryos, making changes that are inherited, passed down through all future generations. On that, she's called for a global pause, a moratorium, warning us that we just don't know the consequences. And this is really the central question we all have to face now. Curing disease seems like a no-brainer, but what about enhancement? What about changing the very fabric of our species' evolution? It's a debate that's way bigger than just science labs. It involves every single one of us. And of course, whenever there's a revolutionary, multi-billion-dollar technology, what happens next? A fight. A really fierce, high-stakes patent battle blew up between UC Berkeley and the Broad Institute. At its core, the fight was over who really deserved credit for being the first to show that this bacterial system could work in our own complex cells, which was the key to unlocking its massive medical and commercial potential. So, you win the Nobel Prize, you spark a global revolution. What do you do next? Just retire? Not Jennifer Doudna. She's working right now to guide the future of the very technology she helped bring into the world. Oh yeah, her work is far from over. Today, she leads the innovative Genomics Institute, this amazing center that's dedicated to using CRISPR to solve some of humanity's biggest problems. And they're not just looking at human diseases. They're trying to create crops that can withstand climate change and engineer microbes that can help heal the planet. In the end, Jennifer Doudna's discovery handed humanity an almost godlike power. We are no longer just people who can read the genetic code, we can now be its authors. The question isn't if we can do it anymore, the question is should we? The story of our future is now, quite literally, in our hands. So what story are we going to choose to write?